Okay, greetings everyone. I welcome you to the third part of our neuroanatomy series, where we're going to look at the basal ganglia. So we have already covered two topics so far. The first one was the introduction to the neuroanatomy series, and the second one was it has to do with the blood supply to the brain. But this time around, now we're going to focus on the basal ganglia. Yeah, you can go to the next slide. Okay, so our objectives for today will be, one, we're going to focus much on the structure and the nuclei of the basal ganglia. From there, we're going to look at the connections or the pathways or the circuits of the basal ganglia. Then after, we're going to focus much attention on the functions of the basal ganglia. And lastly, we're going to look at the clinical relevance of the basal ganglia. So what is the basal ganglia? Basal ganglia is simply a subcortical mass of gray matter that is found in the cerebral hemisphere. So what does this mean? It means we have to break this down. So we have the basal and the ganglia. The ganglia is a group of cell bodies which are located outside the central nervous system. By this time around, the cell bodies we are looking at are those which are found within the central nervous system. So it is also called the basal nuclei. Why? Because basal nuclei are those group of cell bodies of neurons which are located within the central nervous system. So this name is more accurate as compared to the basal ganglia in a sense that ganglia is outside the central nervous system, but nuclei is within the central nervous system. But in this lecture, we're going to use the term basal ganglia. So it is a unique area of the brain which has got high demand for oxygen. So the basal ganglia is not just one component. It comprises of many nuclei that we're going to see in the next slides. So it is the accessory motor system, just like the cerebellum. The basal ganglia receives most of their input signals from the cerebral cortex itself and also return almost all of their output signals back to the cortex. So in this case, the basal ganglia acts more like the control center, or so to say, the part of the brain which modulate motor activities. So what are the general functions of the basal ganglia? So the first we're saying is the part of the extrapyramidal motor system. And secondly, we are saying it facilitates behavior and movement, which is required and it is appropriate. It also limits unwanted and inappropriate movements. For example, there are times where maybe a lecturer is lecturing in class, then you're asked a question, you have to raise up your hand voluntarily. So if you have an answer to the question that is asked, you raise up your hand. And once you're done, you drop your hand back. So there are two things that are involved. There is that initial movement of the hand up, and there is that lowering of the arm down. So we are saying this basal ganglia is responsible for initiating such movement and also stopping the movement. We're going to look at the example further later on as we're going to look at the Parkinson's disease. So the last and not the least function is that of motor control, or the final command pathway, as we will see. So now we're going to look at the nuclei or the basal ganglia. So as we said, the basal ganglia is not just one thing. Yeah, it is a group of nuclei which are located within the cerebral hemispheres. Now, what are these nuclei? So we are saying on each side of the brain, each ganglia has these five components or five nuclei. So we have the caudate nucleus, the putamen, globus pallidus, substantia nigra, as well as subthalamic nucleus. But it is important to note that globus pallidus has two parts. So we have globus pallidus internus and globus pallidus externus. So the basal ganglia is located lateral to and surrounding the thalamus. So we're going to see this in the next few slides, how this anatomical relations is. So it occupies the large portion of the interior regions of both cerebral hemispheres. Since we said it is found on each side of the brain, it means on each side of the hemisphere, we have that component of the basal ganglia. So this is a simple illustration of the nuclei or the basal ganglia. So as we can see, the blue part just on the central aspect, we have the thalamus. Then just on the peripheral, we have the globus pallidus. We also have the putamen, as well as the substantia nigra. So if we are to zoom in, we're going to find that the caudate nucleus is C-shaped, as it can be seen there. So we're able to see the caudate nucleus as C-shaped. Then on its head, more like, not really the head, but the large portion on the end, we have the putamen. So we're saying the putamen and the caudate nucleus, these two, they make up the corpus strata. Then we have the thalamus, which is still in blue. Then we also have the substantia nigra, which we can see in purple down there. Yeah. You can move to the next one. Okay, so this is another view if we are to see it from the posterior aspect. So you can see in green the thalamus, then in red we have the stratum. Now remember we said the stratum comprises of the caudate nucleus as well as the putamen. Then we also have the substantia nigra here and the globus pallidus. So from the globus pallidus, you can see that there are two. There is one which is too close to the thalamus, and the other one which is not so close to the thalamus. So these are the two parts we mentioned to say there is globus pallidus internus and globus pallidus externus. So we are going to look at the functions of these individual nuclei or parts of the globus pallidus in the next few slides. So, how does the basal ganglia work? The basal ganglia works with the use of neurotransmitters. So, neurotransmitters which are at play in the basal ganglia system, we have dopamine, GABA, glutamate, acetylcholine, as well as many other neurotransmitters which originate from the brainstem. So such neurotransmitters such as norepinephrine, serotonin, as well as enkephalin. So, what is the function of this pathway, or rather, where do these neurotransmitters originate? So the pathway that is from the substantia nigra to the caudate nucleus and the putamen make use of the dopamine, as we shall see in the depictions that have been put in the next slides. So we also have GABA, which is responsible for pathways from the caudate nucleus and the putamen to the globus pallidus, and substantia nigra. So when we say globus pallidus, we still mean those two, globus pallidus externus and globus pallidus internus. Glutamate. With glutamate, just like acetylcholine, it plays a role in pathways from the cortex to the caudate nucleus as well as the putamen. Now, among these neurotransmitters, we have those which are inhibitory and those which are excitatory. So what are excitatory neurotransmitters? These are neurotransmitters which are able to cause depolarization of a nerve so that it opens up, and then also have inhibitory neurotransmitters. So inhibitory neurotransmitters, we are saying those neurotransmitters which cause hyperpolarization. So we'll look at hyperpolarization and polarization later on within this lecture. So this is the basal ganglia. The basal ganglia, it has got pathways. So we have the direct pathway as well as the indirect pathway. So there is another pathway which is not put here, and that pathway is called the intrinsic pathway. But here we're going to focus much on the indirect as well as the direct pathway. So where do these pathways begin? So there are two points where we can have the starting point. So first, we're going to focus much on the cortex. So cerebral cortex, it has got many, we have things such as the somatosensory areas, we have precentral areas and others. So that's where we're going to have the initiation of nerve impulses. So the cerebral cortex, so neurons from the cerebral cortex will release glutamate. The GLU we are seeing there is glutamate. Now, as we said, glutamate is an excitatory neurotransmitter. So that's why we have a plus there to show that it is going to cause excitation of both the caudate as well as the putamen, which are collectively called the stratum. So once glutamate activates the caudate and the putamen, but for the sake of understanding for now, we're going to use the putamen on the stratum. So glutamate released from the cortex is going to excite the putamen. The putamen to globus pallidus internus, we said they make use of GABA. And what is GABA? GABA is an inhibitory neurotransmitter. So once glutamate excites the putamen is going to produce neurotransmitters which are going to inhibit the globus pallidus internus. So we can see there GPI, which is the globus pallidus internus. Then we also have the substantia nigra reticularis. Yeah. So it's subthalamic nucleus reticularis. Okay, I'll clarify that later on. After GABA has inhibited the globus pallidus internus, which also produces GABA, it means the globus pallidus internus is going to produce less GABA. And if less GABA is produced, it is going to disinhibit the inhibition on the thalamus. I'll repeat that again. So we are saying the putamen produces GABA. GABA is going to act on the globus pallidus internus. It is the globus pallidus internus which is going to produce GABA, which actually inhibits the thalamus. But now imagine this time around we have an inhibition and an inhibition. So what will happen to the thalamus if we have an inhibition and an inhibition? So if an inhibition is followed by an inhibition, then we're going to have disinhibition. Just like in mathematics, negative times negative gives us positive. So it means here, inhibiting the globus pallidus internus will cause and lead to production of GABA. And if little production of GABA is produced, we're going to have an excitation of the thalamus. Once the thalamus is excited, it is going to produce an excitatory neurotransmitter, which is glutamate. So glutamate is going therefore to excite the cerebral cortex. So once the cerebral cortex is excited, now we can have that initiation of motor activities. Let's take for instance, we have a boy sees a dog, and this boy is scared of dogs. What will happen is the cerebral cortex will release this glutamate. Glutamate is going to act on the putamen, which is going to produce GABA. And once GABA is produced, it's going to inhibit production of GABA by the globus pallidus internus. Once that happens, we are exciting the thalamus. So exciting the thalamus will cause glutamate, which is an excitatory neurotransmitter that will excite the cortex. As a result, the boy will be able to turn and run away from that stimulus. So that is the direct pathway. Now, before we go to the indirect pathway, we can look at the substantia nigra, which is on our right. So that substantia nigra, it is part of the basal ganglia, which produces dopamine. So dopamine is going to be released from this substantia nigra. Once it is released, it is going to act on D1 receptors. The D1 receptors are located in the putamen. So the putamen will again be going to be excited. So there are key things here that I want you to understand. We have D1 receptors. We also have D2 receptors just within the putamen. So where we have D plus, DA plus, it means here this dopamine is going to act on the excitatory receptors, which are called D1 receptors. Then this D minus on the other side, where we have on the left hand side, where we have DA negative. This side, dopamine is going to act on the inhibitory receptors, which are called D2 receptors. So these two receptors are very important for us to understand the two pathways. Now, back to where we were. So I'm saying dopamine is going to act on D1 receptors on the putamen. Again, the putamen is going to be excited so that it produces more GABA, and the process continues. So GABA will act on the globus pallidus internus, and this little or no GABA is going to be produced. As a result, we are going to have excitation of the thalamus. Once that happens, what will happen is we will have excitation of the cortex. So that's the direct pathway. Do you have any questions on the direct pathway before we go to the indirect pathway?
No, doc, it's very clear.
All right, thank you. So now we're going to look at the indirect pathway. So the indirect pathway, this time around, we want to inhibit the cortex. Initially, we wanted to excite the cortex. By this time around, we want to inhibit the cortex. So that's the major difference between the indirect pathway and the direct pathway. The indirect pathway is involved in inhibiting the cortex, while the direct pathway is involved in the excitation of the cortex. So again, the cortex will still produce glutamate, which is going to act on the putamen or the caudate nucleus. So this time around, let's use the caudate nucleus so that we don't just leave it. So glutamate is going to act on the caudate nucleus. Once that happens, there will be production of the caudate will produce more GABA. Once more GABA is produced, it is going to act on globus pallidus externus. So this time around, we are not looking at the globus pallidus internus, but externus. Just as we said earlier, say these two parts of the globus pallidus have got different roles. In that, globus pallidus externus is involved in the indirect pathway, while the globus pallidus internus is involved in the direct pathway. So after GABA has acted on the globus pallidus externus, the globus pallidus externus is also going to produce GABA. Now, let's say this time around, this globus pallidus externus is going to produce less GABA because it has been inhibited by the previous GABA from where? From the caudate. So once that happens, it means we're going to have less inhibition on the on the subthalamic nucleus. So the STN here is the subthalamic nucleus. So once that happens, the subthalamic nucleus is going to produce more glutamate. Now remember, glutamate is an excitatory neurotransmitter. And if it is an excitatory neurotransmitter, we're going to have exactly the opposite. So if it acts on the globus pallidus internus, which we said produces GABA, it means we're going to have more GABA released. And once more GABA is released, it's going to act on the thalamus. Now what will happen? The thalamus will produce little glutamate. So once that happens, we are inhibiting the cortex. So that's the indirect pathway and the direct pathway. If there are questions on the indirect pathway, you can let me know so that we can move to the next if there are no questions.
Okay. It's clear. You can proceed.
All right, Doc. Okay, so this is just a simplified model to illustrate what we said. So here we've just extracted the direct pathway where we said the cerebral cortex is going to produce excitatory neurotransmitter, in this case, which was glutamate. And the glutamate is going to act on the stratum. Remember, in the stratum, we have the caudate nucleus as well as the globus pallidus, as well as the putamen. So once that happens, these two guys will produce inhibitory neurotransmitter, which is GABA. And the GABA is going to act on the globus pallidus internus. And the substantia nigra pars reticulata. So just go to the previous slide. Yes, so here where we have globus pallidus internus and SNR, here where I said we'll classify it later, SNR is the substantia nigra pars reticulata. You can go to the next slide. So once that happens, these two guys will also going to what? To release more to say less GABA. Since they have been inhibited, so this X shows that there will be little or no GABA that is going to act on the thalamus. So if that happens, it means this thalamus will have more glutamate being produced in order to excite the cerebral cortex. You can move to the next. Okay, so this is just another way of representing what we said. It all starts with the cerebral cortex and back to the cerebral cortex again. So just here, I want you to take note of the D1 receptor and the D2 receptors. So these two receptors are the ones that we were saying to say we have an inhibitory inhibitory receptor as well as the excitatory receptor. So us activating the D1 receptor, it means we are going to more likely excite the stratum, and that will lead us to the direct pathway. But exciting the D2 receptor, which is an inhibitory receptor, it is going to cause inhibitory actions. As a result, we're going to have an indirect pathway which leads to inhibition of the cerebral cortex.
Next.
Okay, so here I was supposed to put the indirect pathway, but I'm still working on it.
Okay.
Although it has already been understood, we can go to the next. Okay, so within the basal ganglia, we have neuronal circuits. So these neural circuits are the most two important ones, are the putamen circuit as well as the caudate circuit. So we're going to look at these two individual circuits and look at some of the diseases that are associated with their lesions. So with the putamen circuit, we're saying this one controls the complex patterns of motor activity, e.g. learned skills. So what are some of the learned skills that we know? So some of the learned skills that has to do with motor activity has to do with essay writing, the handwriting. Normally we maintain our handwritings. Why? Because those are learned motor skills. Things like cooking, things like, let's say, playing football, things like playing volleyball and stuff. So those are learned skills. And the circuit that is responsible for that is the putamen. So it has input from the pars and parts of the brain adjacent to the primary motor cortex. I think earlier on when we are looking at the anatomy of the brain, which was presented by Dr. Mkambo, he showed us where the primary motor cortex was. So now we're saying the inputs that are actually going to activate the putamen arises from that primary motor cortex. Now, if we look at the output, they usually go back to the same primary motor cortex or closely associate itself with the premotor as well as the supplementary cortex. Now, what happens if the putamen circuit is altered or if something happens to it? The way the putamen execute patterns of movement is poorly known. However, when a portion of the circuit is damaged or blocked, certain patterns of movement become severely abnormal. Now, what are some of these movements that become abnormal? So we have lesions of the substantia nigra, which leads to common and extremely severe disease of rigidity, akinesia, tremors. And these three are classical signs of Parkinson's disease. So we are saying the substantia nigra, as we said, it produces dopamine. So imagine we don't have dopamine or we have less dopamine. It means we are inhibiting which pathway? Of course, we're inhibiting both the indirect and the direct pathway. The direct pathway will be inhibited in situations where we have, let's say, the cortex doesn't produce enough glutamate. So we rely much from the substantia nigra. So it means the excitation of the cortex is going to be altered. As a result, we're going to have difficulties in making movements. We're going to have tremors, especially resting tremors, as well as rigidity. So the second lesion occurs the globus pallidus. And this lists spontaneous and continuous breathing movements of the hand, the arm, the neck, as well as the face. So if this happens, they lead to a condition that we call athetosis. So athetosis is simply the continuous breathing movements of the hand, the arm, the neck, as well as the face. Lesions in the subthalamic nuclei often lead to sudden flailing movements of the entire limb, and this we call hemiparesis. So hemiparesis, we can have paresis itself, which involves both sides of the body. We also have hemiparesis. So how does hemiparesis comes about? We'll look at it in detail once we start looking at the clinical correlates. So we might also have multiple small lesions in the putamen, which lead to flicking movements in the hands, face, as well as other parts of the body, and that is called chorea. So we'll look at this more as we look at the clinical correlates. Now, other than the putamen circuit, we also have the caudate circuit. Remember on the stratum, copper stratum, we had two parts. So we had the putamen as well as the caudate nucleus. So if the circuit involves much of the caudate, we're going to call that as a caudate circuit, and vice versa with the case of putamen circuit. The caudate circuit plays an important role in cognitive control of motor activities. It receives a large amount of input from association areas of the cerebral cortex overlying the caudate nucleus. So signals return back to the prefrontal cortex, premotor cortex, and the supplementary motor areas of the cerebral cortex. But with most of or none of the returning signals passing direct to the primary cortex. So it also plays a role, or the role is appreciated when seeing a lion, which is a similar example that we gave with the case of a boy fearing a dog. So the circuit that plays a role in an example such as this one. So we have a lion. Let's say you see a lion, then the response that will follow is, of course, you're going to turn away from the lion. You begin to run, and you even attempt to climb a tree in order to stay away from the lion. So all these are connected through the caudate circuit. So that's the major function of the caudate circuit. So now we can look at the clinical syndromes from the basal ganglia damage. So what happens if there is a lesion to part of the basal ganglia? Could be part of the. So we have these major three syndromes or diseases. So we have Parkinson disease, Huntington disease, as well as the hemiparesis. Now, under normal circumstances, we have the direct pathway and the indirect pathway working normally. But in Parkinson disease, the main thing we're going to focus much on is down there where we have the substantia nigra. So if there's a lesion in the substantia nigra, we're going to have Parkinson disease. Then we come to Huntington disease. So Huntington disease, normally, what happens that we have defect in the genes, which are called Huntington genes. So those genes are responsible in coding specific proteins, and we're going to look at this in detail later on. Then we also have hemiparesis. Hemiparesis, where we said there is a defect as we said in the previous slides, where we said if the defect involves the subthalamic nucleus, we're going to have hemiparesis. But we are going to look at the mechanism by which this disease comes about in the next slides. So we'll start with the Parkinson disease. So Parkinson's disease is also called paralysis agitans. So there is usually destruction of portion of substantia nigra, which is pars compacta. Remember earlier on, we said we have substantia nigra pars reticulata and substantia nigra pars compacta. But this time around, for us to have Parkinson's disease, normally, neurons which are located specifically in the pars compacta or the substantia nigra are usually damaged. As a result, we have deficiency of dopamine, which is going to affect the indirect and the direct pathways that we described previously. So what are some of the features of Parkinson's disease? So as we said earlier on, there were three major ones where we said rigidity, akinesia, as well as we have tremors. But normally we have what we call resting tremors. Now, we can have akinesia, which is heavy, heavy difficulties in making movements, or we can have bradykinesia, which is usually we can have moderate to severe difficulties in making movements. So the cause of abnormal effects of movement is not much known. So we can look at the treatments of Parkinson's disease. How can we treat Parkinson's disease? So you can treat Parkinson's disease, one, by administration of levodopa, which is usually abbreviated as L-dopa, and carbidopa. So dopamine itself, we're saying it won't work. Why won't it work? We're going to see in the next slide as well. So we're saying L-dopa and carbidopa are usually administered at the same time for the reasons that we are going to see just in the next slide. Administration of drug L, deprenin, which is a monoamine inhibitor. So we're going to look at where this monoamine comes in. Then we have treatment with transplanted fetal dopamine. So if we are to do this, it's more like you want to replace the dopamine function there. Because we say Parkinson's disease usually has to do with the deficiency of dopamine due to damage to pars compacta of the substantia nigra. So we have also treatment by destroying part of the feedback circuit in the basal ganglia. So I say there is okay, we'll see this in the next depictions. Yeah, you can go to the next one. Now, we said levodopa is usually given along with carbidopa. Why is that the case? So I'm saying levodopa, I think you can see here, it has to be converted to dopamine. Now, there are two things here. We have the peripheral as well as the central. Now, the dotted line here we are seeing it is the blood-brain barrier, BBB. So, now for dopamine, dopamine can't cross the blood-brain barrier due to its molecular weight. But levodopa usually crosses the blood-brain barrier without much difficulties. So according to a research, they said if we administer levodopa alone, just about 1% of it get its access to the central, or meaning gets to pass through the blood-brain barrier. But if we are to administer it with levodopa, we're going to have at least 10% of it crossing the blood-brain barrier. Now, we don't want dopamine outside the blood-brain barrier because it has got several side effects. So, as administering it with carbidopa, we are simply inhibiting the conversion of levodopa to dopamine. As you can see the negative there, it simply shows that levodopa can't be converted to dopamine if it is outside the blood-brain barrier. Now, if we get entry into the blood-brain barrier, we find that levodopa is converted to dopamine. And inside the central nervous system, we're saying carbidopa facilitates the conversion of levodopa to dopamine. Another thing that we have to note is that here where we are saying the X there, so there it means carbidopa and dopamine, these on their own, they can't get entry to the blood-brain barrier. So once this happens, at least now we have enough dopamine in the substantia nigra, which will be able to, carry on with the pathways that we discussed. So again, this is just to show us some of the drugs that are used in the treatment of Parkinson's disease and where exactly they act. So we have in the circulation, as well as here inside the brain, or inside, so to say, the blood-brain barrier. As we can see the blood-brain barrier there. Yes, so we can see the blood-brain barrier, and we have the circulation in blood just outside, as well as inside this side where we have these neurotransmitters. Now, we have already discussed this to say levodopa is going to cross the blood-brain barrier. Once it crosses the blood-brain barrier, it is going to be converted to dopamine. Now, inside, we don't want dopamine to be converted to monoamine blockers, monoamine B or MA or B. And we don't also want dopamine to be converted to 3-OMD. So I'll write this in the next slide as psych-on editing, the meaning of these words. So for now, let's just take them as the more like other substances that dopamine can be converted to in the brain. So we are preventing this from happening. We want dopamine first to do its role before it can be converted to these two. So we have monoamine B inhibitors, which are, if you are to zoom in, help me zoom in, Doc. Especially where there is the monoamine B inhibitors.
Sorry, give me a second.
All right. Okay, just like that. So if we are to zoom in here, we're going to see selegiline as well as rasagiline. So these are two drugs which are able to prevent this conversion. So this is in the presynaptic neuron. So we want dopamine first to cross so that it can go to the postsynaptic neuron. Now, before it can cross, there are possibilities that it can be converted to AM or B. So we have to administer drugs which are going to cross the blood-brain barrier as well as inhibit this conversion. Similarly, dopamine can be converted to 3-OMD. Now, we have tocapone. The tocapone is the drug that will actually inhibit this conversion. So, if we have these three agents that are acting to prevent dopamine from being degraded, then we're going to have the dopamine into these neurotransmitter vesicles, which is going to cross the presynaptic neuron and get entry into the postsynaptic neuron. Now, what will happen here is just the binding to the dopamine receptors. As we said, let's take for instance this neuron, which is on the postsynaptic, is the one now uh, on the stratum. Now, stratum specifically, we are talking about the putamen. Let's say this neuron is in the putamen, and the other neuron, it is coming from the substantia nigra. So substantia nigra has released these neurons so that they can bind on the dopamine receptors, either D1 receptors or D2 receptors, as we discussed earlier. Now, again, there's another agent that we can use in order to promote or increase the availability of dopamine just within the gap in between these two neurons. And that agent is right there above amantadine. So amantadine is the drug that we can use actually to promote or to increase dopamine in here. Again, we have dopamine agonist. So what is an agonist? An agonist is simply a drug which promotes more like the entry of another drug, or so to say, an agent which promotes the existence of another agent. So in this case, these dopamine agonists are going to bind to dopamine receptors so that they open more and more dopamine can get entry to the postsynaptic neuron. This time around, could be in the caudate or the putamen. So this was just to show us some of the drugs that we can use to treat Parkinson's disease. So having done with Parkinson's disease, we can look at Huntington's disease. So Huntington's disease is a hereditary disorder. It usually begins causing symptoms at the age of 30 to 40 years. At first, flicking movements occur in individual muscles, and then progressive severe distortion of movement of the entire body. So severe dementia along with the motor dysfunction may develop. As we said, the Huntington disease, it is usually due to defect in the Huntington gene. So you can go to the next. Okay, so what are some of the causes of Huntington disease? So these some of the causes include loss of most cell bodies of the GABA secreting neurons in the caudate nucleus and the putamen. So as we discussed earlier on when we are looking at the putamen circuit, we noticed that damage to the caudate nucleus and putamen will actually one of the predisposing factors to having Huntington disease. So I'm saying if we have not enough cell bodies that produce GABA in those two nuclei, that is the putamen and the caudate, it means we're going to have less GABA being produced. And if less GABA is being produced, what will happen? I just want you to go to the previous slide where we had that pathway which was showing GABA. Yeah, just here. Now, imagine we have defect in the stratum, specifically in the putamen and the caudate. So here, if we don't have enough GABA, let's concentrate first on the indirect pathway. I mean the direct pathway. Sorry. Yes, okay, just here. It's okay. Yeah. So if we focus here on the direct pathway, let's take for instance the putamen. If the putamen doesn't have enough cells which produce GABA, what will happen? We're going to have less GABA, and if we have less GABA, it means we're going to have less inhibition of globus pallidus internus. So once we have less inhibition, what will happen? The globus pallidus internus is going to produce more GABA. And what happens if more GABA is produced? Then we're going to have a much increase in inhibition of the thalamus. Now, what happens if the thalamus is inhibited? Of course, what will happen is that we're going to have less glutamate being produced. If less glutamate being produced, then we're going to alter the direct pathway. So in short, the cortex won't be activated, more like to enhance the required motor activity. Again, if we focus much attention on the indirect pathway, let's say we still have less GABA being produced from the caudate nucleus. Once less GABA is produced, it means there will be less inhibition of the globus pallidus externus. Once that happens, globus pallidus externus is going to produce more GABA. So that more GABA is going to have much influence in inhibiting the subthalamic nucleus. So once that happens, we're going to have less glutamate activating the globus pallidus internus. So if that happens, then we're going to have less GABA acting on the thalamus. I mean, we're going to have less GABA, yes, so to say. So we're going to alter the indirect pathway. Let's say the movement wanted is to, let's say